Hi, my name is Sadie Beal, and I'm a Sports Medicine 3 student at South Tahoe High School. For my fourth project, which is medical journal research, I chose to research diffractometers and how they're used to study the water dynamics of the brain. I'll start off by discussing why my topic is important. The human brain is one of the world's most fascinating mysteries, with its inordinate intellectual capacity and ability to do advanced computations and communication. But the study of the brain is limited due to the fact that options like X-ray diffraction and imaging can be extremely harmful to the brain. However, neutron scattering is proven to be risk-free for the brain and shows promising results about how the water dynamics and anomalies in the lateralization of brain tissue. The neutron scattering process uses a 3-axis spectrometer and a diffractometer. It is it produced results that give information about the myelin sheath structure and how water interacts with cell membranes. This will further help medical research on tumors, brain diseases, and more due to the study on anomalistic lateralization that occurs during diseases. Now I'm going to explain what the basics of diffractometers are. Diffractometers are instruments used to analyze and study materials. If you didn't know, diffraction is a modification of light which light undergoes essentially in passing by the edge of opaque bodies or through narrow openings in which the rays appear to be deflected. Diffractometers utilize the scattering pattern that is produced when a beam of particles interacts with the material. For the type of diffraction I am studying, the particles used are neutrons. Neutrons are better than electrons or radiation beams because radiation beams can be harmful and electrons are only able to analyze superficial structures. Next, I'm going to introduce water dynamics and the study that I researched. The brain is made up of heterogeneous systems that each contain different types of compartments, which are separated by impermeable and semi-permeable and semi-permeable membranes. More than 70% of the brain is made of water, and there are psychological differences in the water content between various brain regions. This water plays a key role in regulating ion concentration gradients across membranes, which is vital for the conduction of nerve impulse. When water molecules interact with cell membranes during random motion, it can be used as a tool to probe tissue and therefore provide researchers with information about it. One way to do this is with neutron scattering, a space-result spectroscopy technique. Neutron scattering is one of the best techniques because the probe distance can be reduced to the scale of macromolecular separation. Unlike radiation waves, the neutrons don't have a damaging effect on the brain, and due to its incoherent cross-section, the nucleus scattering signal is sensitive to hydrogen nuclei, which makes it ideal for studying water dynamics. It allows scientists to research proton dynamics at an atomic scale, no matter the complexity of the macromolecules. The neutron scattering spectroscopy has allowed scientists to get a better understanding of the water dynamics in various biological environments and allowed them to gain information on atomic molecular dynamics on the picosecond timescale, which is essential to understanding various biological functions. One technique that is often used is quasi-elastic neutron scattering, which I won't be discussing much in this presentation, but is really interesting because it allows researchers to measure molecular displacement in time and space at the picosecond angstrom scale, and is able to describe the specific motions of hydrogen in different water pools in the brain in order to compare the properties of water in different cells. In this study, researchers applied new neutron scattering techniques and gained insight on how it can be used to study the water dynamics of the brain. For the first experiment I researched, you have to begin by using a cryotome to cut 50 micrometer thick slices of hypothalamus, which is the part of the brain that controls body temperature, hunger, and thirst. The thickness was chosen to limit neutron absorption from water, which minimizes the chance of multiple scattering events taking place. The sample was then closed in a vacuum-tight pure aluminum container. The experiment was then initiated at room temperature on a small momentum transfer diffractometer, which was used at a wavelength of 4.2 angstroms. This device, also called an ion 14, is a neutron 3-axis spectrometer at an institute in France. After the sample was in place, an incident cold neutron beam was diffracted on a PG002 monochromator in order to create a monochromatic beam, and a nitrogen-cooled beryllium filter was installed before the sample in order to reduce contamination from the monochromator. The beam was collimated before and after in order to ensure an accessible region of momentum transfer Q down to 0.06 angstroms. Another experimental research was conducted at room temperature in a small momentum transfer diffractometer. This device, also called a D16, is a diffractometer that uses a neutron beam from a graphite multidetector, which is used to study structures of 50 angstrom, periodically diffracting to a real space resolution of a fraction of a nanometer. In order to enhance coherent signal, the sample was left in a deuterium oxide atmosphere for half an hour. The monochromator delivered a wavelength of 4.75 angstroms at full width half maximum. The distance from the sample to the detector was set to 950 millimeters. The collected data was integrated vertically. At a range of negative 1.2 degrees is less than 2 theta vertical, which is less than 2.8 degrees. The first results from the study I have are graphs. This is figure 1. These are the mean diffraction patterns of the D16 diffractometer and ion 14 at 300 degrees Kelvin. This graph shows relationships between Q, which is momentum transfer in angstroms, and diffracted intensity. This is figure 2a. The graph shows similar data to figure 1, however it separates the intensity in the left hemisphere and the right hemisphere. This is figure 2b. The figure shows the myelin sheath structures, which was concluded from figure 1 and figure 2a graph. The repeat distances correspond to the Bragg peaks on the graph. As a result of the experiments, details were concluded about the lamellar myelin sheath in the brain. The diffractometer D16 measures neutron diffraction patterns at 27 degrees Celsius or 300 degrees Kelvin, which corresponds to figure 2a. The position of the Bragg reflections help deduct the associated repeat distances, the d-spacings, and the myelin structure in figure 2b. The shift in the graph from the left hemisphere Bragg reflections and the right hemisphere Bragg reflections suggest different myelin sheath arrangements between the two different regions in the brain and a non-negligible change in thickness and density of sheath. Figure 1 showed similar data, where the peaks of Q are 2 pi over d, where d is the repeating distances. Overall, these graphs have indicated conclusions that the water in the right hemisphere has more free range than the restricted water in the left hemisphere on both micron and atomic scales. Results from the neutron scattering techniques in the right hemisphere of the brain displays that there are two different water groups due to the contrast in the dynamics and behavior. The heterogeneity in the dynamics connects back to the cellular constituents that hinder diffusion of water molecules. The water diffusion is jump-like in its multiformity in the various subregions because of interactions with the cell membranes. To conclude, neutron scattering is proven to be one of the only promising scans that does not harm the brain. Through methods like the 3-axis spectrometer and the small momentum transfer diffractometer, crucial information about cell structures, water dynamics, and how the two work to create the brain was acquired. The experiment started off by using a neutron beam for scattering, using specific wavelengths that gave information for the graphs, which represent the direct correlation between momentum transfer and intensity. This has allowed researchers to infer many things about the water diffusion in the brain and calculate despacings in the myelin protein structure. Researchers can use neutron scattering experiments to take a closer look at the differences in brain conductivity and help understand how the brain lateralization contributes to the brain function. These experiments can help researchers gain more information about the abnormalities that differ from the standard water diffusion, and those abnormalities can help with research for brain diseases. Here's my works cited, and I also know a lot of this information because I've had to do a lot of research on this in the past. Thank you.